Today's topic, we're going to look at the two processes that you actually already did at EcoLink, which is the polymerase chain reaction and the gel electrophoresis. Okay, you guys have all of you did it, right? Who was not that? Yeah. 4B, which is CRISPR, is too big of a content to cover in this class right before the end of term, so I'll be doing the video on that. So one of the topics I'll be covering on the video is 4B, which is CRISPR, which is honestly a very big component of your entire Unit 3 and Unit 4 because it's mentioned multiple times across the two units. So I'm going to take a bit more time to make sure we cover the content properly for that one. For this one, you guys have already done it at Ecolink, so you already know on some level how it's done. So that's why I'm going to finish off the term by just doing this because I don't expect you guys to be running at 100% right now. All right. So today, the design that you are expected to know is how chain reaction is used to amplify DNA and how gel electrophoresis is then used to sort DNA fragments based on their sizes, and then that is used as an interpretation method for DNA profiling. So essentially, if you've ever asked yourself how detectives or CSI has ever been able to use DNA to match suspects, it's using this one term of DNA profiling. Okay, they're essentially using two techniques in the simplified way, which is the polymerase chain reaction and gel electrophoresis. You can conduct uh, basically DNA profiling to identify individuals. Okay, so some of the expectations are that you guys are able to define and understand how polymerase chain reaction works. Second is explaining the steps involved. Third is the how to make up a gel for gel electrophoresis. And then most importantly out of that is actually knowing how to read the gel, because a lot of questions will just give you the picture of the results of the gel. You need to be able to decipher information based on that. If you do not know how to read a gel properly, all you're going to see is a complete gibberish in the picture. So that's why it's very important that you actually very much understand how to read a gel first, and then only we can work on interpreting it. Okay, so let's do some review regarding the enzymes that manipulate DNA. All right, Yasmin, what is a type of enzyme that cuts DNA? Restriction endonuclease. Good one. Okay. Another way you could refer to them is restriction enzymes, if that's easier for you to remember. Both names are interchangeable. There's nothing wrong with using one or the other. I prefer to use restriction enzymes because it's just easier to remember that way. Okay. So next question. What are the two types of cuts that restriction enzymes can make? Can I get Mare? Okay, there's two types of cuts a restriction enzyme can make. One is a kind of cut that can attract, and the other is won't have any attraction. All right. Anyone want to help out? Gavin, what are the two types of cuts? The name very much explains what type of properties it has. Abi, what do you think? Really? Wow. Evelina, any ideas? What are the two types of cuts restriction enzymes can make? I'll give you a hint. I'll draw it out for you. Okay? If this is a full DNA, right? Two strands. Okay. One type of cut will be like this, where it leaves an overhang, right? Sticking out, where there's nucleotides sticking out that can bind. Another one is where the cut literally makes a perfect incision to cut the DNA right between the restriction site. Okay. So what are these two cuts? Ayman. Sorry? And you. Sticky. So yeah, this is a sticky end and this is a blunt end. Okay, sticky end because it has nucleotides hanging, which act as an attraction for the correct type of DNA strands in the correct direction. Whereas with blunt ends, there's no attraction, so it is less precise in what it does. Okay. Next question: what is an enzyme that makes functional DNA strand? Okay, you guys have already learned about this enzyme. It makes functional RNA. So this one I'm asking which one makes a copy of a functional DNA. All right. Can I get Yasmin? Oh, I already asked you, right? Janita. Okay. Think about what makes a functional mRNA copy. So what would make a functional DNA copy? It's not too complicated. RNA polymerase. Yeah, that's the answer. It is literally the same words. All that's different is instead of generating RNA, it's making DNA. So you already know the enzyme that makes functional nucleic acid. You just have to change the name based on what type of nucleic acid it makes. One is RNA polymerase, the other one is DNA polymerase. Okay, it's making functional DNA strands. That is a DNA polymerase. If it's making mRNA copies, it's a RNA polymerase. Okay, always ask yourself what type of strand it's making, and you'll know what type of polymerase is in charge. So, this okay, the function of DNA ligase for this during the preparation of the recombinant plasmids, researchers use the enzyme DNA ligase. Okay, so we've looked at restriction endonuclease or restriction enzymes, then DNA polymerase or RNA polymerase. Then the third enzyme that we looked at for manipulating DNA is DNA ligase. What is the function of that, Zoe? What do you think? Yeah, so which one do you think would be the most accurate answer here? Yep. There you go. Right. B. Join the disruptin G to the plasmid DNA at complementary sticky ends. Right. So essentially, anytime you see a gene or a segment of DNA being joined together to a plasmid or another copy of a DNA, that is done with the action of DNA ligase. The ligase basically glues those two fragments together to make one complete strand. So that is its function. So you can always remember that one. Okay, remember that DNA ligase is about pasting the DNA fragments together. Restriction enzyme is about cutting it, and polymerase is about making copies. Okay, DNA polymerase makes DNA copies. RNA polymerase makes RNA copies. All right. So these are the glossary tasks. As I said, during the holidays, your homework as it is to finish all the coursework. Okay. So first day back, that's the first thing I'm gonna do. We're gonna go through a checklist of making sure everyone's done the coursework. And if they have not, then yeah, there will be what's the word I'm looking for? Consequences. Yes. All right. So DNA essentially is not just about a blueprint to make proteins, to make the cells that make up you. They also act as it can also act as a form of identification. So think of a barcode for the items you buy at a supermarket, right? Each barcode that each item has is a unique barcode, and you can use that barcode to identify them. Same thing with the DNA here, right? The DNA acts as its own barcode that we can then actually read to find out who's who. So this is an example of a simple way gel electrophoresis would look like, right? We can actually see if this is the DNA fragments of the bit of the evidence left on the prime. You can compare it against the DNA fragments from other samples and then find the one that is the most matching with that. And then you have a very good a very good idea that who is the suspected criminal. This kind of evidence is so foolproof that it is accepted in court, right? Even stuff like polygraphs are not accepted in court because there's a lot of give and take. Lie detector tests, they're not always accepted. But DNA tests are even grounds to restart cases at times. So people who are on death row can sometimes even get another chance because new DNA evidence has been found, because that's how strong of an evidence DNA can serve, right? So it is definitely used as a good identification in the 21st century. And even though 99% of the human genome is actually very similar between each other, there is that 1% that is very different between each other. We call these kind of places high variability regions where there's a lot of changes that occur, a lot of mutations. So in between each person, this 1% of regions can have a lot of variability. That is the word we use, right? A lot of differences, and we can use those differences to then identify for the unique tags that each person has. So the two steps to a simple DNA profiling is the polymerase chain reaction and the gel electrophoresis. Of course, the techniques used today can be more advanced than this. This is the most basic level that you guys can actually get away with. And you guys actually already use this in part of your EcoLink, where you actually use polymerase chain reaction and gel electrophoresis to test for a certain gene in the sample that you've collected. So that is exactly how you could use it to identify against other samples, other suspects. So the first one we're looking at is polymerase chain reaction, right? But before we can even do that, it all DNA sample from the suspect, right? That's how any forensic scene starts. They always have to collect DNA from the sample, okay? But here's the thing, not all samples are going to be consistent. Some samples can give a very small amount of DNA to use. Some samples can give a lot of DNA. So to ensure that we can use all the samples regardless of how much copies we have to first amplify those copies, right? Basically make millions more of DNA copies of the same sample that we collected, so that then we can use it for identification. And this polymerase chain reaction, which is a process to make copy DNA strands millions of times, there's three distinct steps. And these three distinct steps are repeated over and over and over. So we call this one cycle. And this machine runs sometimes up to hundreds of cycles even. Okay, it depends on how many copies they want to generate, but the fact is this cycle that you see here is repeated over and over, such that it makes many copies of the DNA strand that is used as a sample. Okay, this process of training millions of copies is impossible without two things. One is the machine itself, which is able to undergo this process in a cycle multiple times. Second is an enzyme. This enzyme is actually an example of DNA polymerase. We call it Taq polymerase because of where it's coming from. That Taq polymerase is actually obtained from a bacteria that is located in hot springs. So that's the reason why we have to use that bacteria, is because it needs to be able to function at a high temperature. So we were looking for a polymerase, DNA polymerase, that can function at a high temperature. That's why we found Taq polymerase, which is from a bacteria that survives in a hot spring. Remember that I've said that enzymes are temperature specific and they can denature very easily, but depending on the living thing that has those enzymes, they can have very different ranges, right? Taq polymerase is an example of an enzyme that can survive, that can still function at a high temperature because it's coming from a bacteria that usually survives at hot springs. So it needs to be able to function at a high temperature. That's why it can function at 72 degrees Celsius. So for a PCR reaction to be able to be done, there are that are needed. One is a primer. Primer is basically something that targets the DNA strand. So you collect a sample, that's the sample you're going to be amplifying. Primer basically is a target that binds that sample to allow for DNA polymerase to create a functional strand from that. Okay, I'm going to go through the steps very clearly after this, but yeah, basically restriction endonucleases are also used in PCR, but they're not always needed depending on the fragment that you're trying to amplify and depending on how they're collecting that fragment. If it's a sample collected from a crime scene, they might not need restriction endonucleases. But if there's a sample that they're trying to analyze, basically they're trying to see whether the sample has this gene or has this segment of DNA, then they might need to use restriction endonucleases. Tag polymerase obviously is needed, and then free floating nucleotides, because for a DNA polymerase to make a functional copy, it needs nucleotides to then join together to form one chain. That's why it is needed that there is all the free floating nucleotides. And then lastly, the machine itself. So let's go through a short video that explains it. Make sure you're paying attention because it pretty much very clearly explains it.
Uses repeated polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
All right, let me go through the video one more time. This time, let me explain each step a bit more clearly.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double. All right, so this is the first step, right? The first step is referred to as denaturation, right? So this happens at a very high temperature, which is 95 degrees Celsius. At this stage, what happens is that the double-stranded DNA will be broken down and the strands, it will be separated into becoming single strands, okay? Only then can the primer attach itself to the strand to make a functional copy, right? Only then can it be free for the DNA polymerase to make a functional copy as well. So this process is always the first, which its primary function is to basically break the double strand to create single strands to facilitate the next step. Okay? DNA to separate or denature into single strands. And then the next step. This is when it's brought down to 55 degrees, right? This is a process called annealing. So the process of annealing is basically just to allow primers to bind to the single strands. Remember that primers, they're basically looking for a specific target. They are sequence specific and they want to bind to a sequence that is complementary to the one that they have. So they are basically targets that look for something along the DNA sequence to bind to Once it's bound, then the temperature is then brought back up again.
Sequences known as fragments. Scale to complementary matches on the target DNA sequence.
Now the temperature is brought back up to
Bracket the target sequence
62 degrees.
at a slightly higher temperature. The enzyme Taq polymerase, shown here in blue.
So remember, like I said, the optimal temperature at which enzyme Taq polymerase is able to function is 72 degrees. So now the temperature needs to be brought up. And this is the final process, and that is called elongation. And this is happening at a temperature of 72 degrees Celsius. Okay, so this is the optimal temperature at which Taq polymerase works at. So once will bind to the primer, where the primer bound. Basically, the primer then also acts as a binding site for Taq polymerase, and from there, a functional copy is made using the free nucleotides that can be found in the solution. Okay.
Sequences and adds nucleotides to extend the second strand.
So remember that the nucleotides, they can be used by the Taq polymerase at any point. That's why they need to be in excess. They basically need to be drowned in nucleotides.
Denaturing, annealing, and extending are repeated.
This process will continue over and over to make more and more functional copies.
After 3 cycles, the target sequence defined by the primer.
So something you notice is that every copy that the reaction creates, that the cycle creates, can then become a template strand for another copy to be made. So this is a exponential growth. Okay, it is not just doubling. Sorry, it is a doubling effect that eventually picks up so much speed that by the 30th cycle, it's already made 1 billion copies. So it is actually a very much an exponential growth.
Begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Alright. Moving on. So yeah, essentially that's it, right? Starts off with the first step, denaturation, separates the DNA strand into single strands, and then the primer binds to the single strand as a target, making sure that it binds to the complementary target, and then the temperature is brought back up to 72 degrees, where Taq polymerase binds to the primer and then continues to elongate the strand, basically make a copy of the strand by using the free nucleotides. And then after elongation, the temperature is brought back up to denaturation levels, which is 95 degrees, where the new copy that was made will denature, causing 2 strands to appear, and primer will bind. This cycle is continued over and over and over. So before I talk about the separate ingredients, yeah, as you can see, the effectiveness is PCR is very high, regardless of whatever amount of copy you started out with, right? Even if you started out with 10 DNA molecules or 5, eventually it will generate enough copies to be able to be used for DNA profiling. So talking about the 4 ingredients, like I said about each other, let me just cover some last details. For one thing, I've talked about how Taq polymerase is an enzyme and that it is also optimally supposed to survive, optimally functional at 72 degrees. But one step of PCR is bringing it up to 95 degrees as well, right? And we know for a fact enzymes don't do well with high temperatures. So luckily, this enzyme has a half-life of 40 minutes at 90 degrees Celsius. So it can actually survive to a certain degree at 90 degrees Celsius, even though it does lose its functionality over time, but it can survive a certain amount of time. So as long as this cycle is continued over and over and not maintained at 95 degrees, it can still function to a certain degree. Okay, next is the DNA sample to be amplified. the DNA primer, basically a dart that looks for its target sequence on DNA to bind to. And then lastly, the free-floating nucleotides. These are all your adenine, guanine, cytosine, and thymine, right? DNA tag polymerase will then use those to make a functional copy. Okay, anyone confused about PCR so far? Okay, any step? Okay, the 3 steps: denaturation, annealing, and elongation. Alright. The next one is the step where they then use the amplified piece to band, as a form of identification through a banding technique. Okay, so gel electrophoresis essentially is a banding technique that uses a few principles to be used as an identification technique. Okay, these principles basically allow gel electrophoresis to do what it does. First thing is that all amplified DNA fragments are cut by restriction enzymes at restriction sites found on the DNA fragment. This is important because considering whatever sample you've gathered, it will have the same restriction sites as the any comparison you're going to use, right? So essentially, how do I explain this? So let's use this one as an example. Okay, right here we have a gel electrophoresis, and these are the sizes of the fragments that are generated. Those are 400 base pairs, 500 base pairs, 300 base pairs. When you look at sample 1 and sample 4, you can see that there's a similarity. Both of them have the same number of bands. Basically, what that means is that the DNA fragment, that the full DNA fragment, there were restriction sites at the same place that were cut to generate the same kind of fragment sizes. So essentially 430 base pairs, 430 base pairs. So this was the same for sample 1 and for sample 3. So that's why you can use it as a comparison. Since the sample and whatever you're using to compare will have the same sites for the restriction enzyme, then you can use that as an identification technique to compare the bands. Okay. This will probably make more sense as we go through it. Let me go through the next principle, which is, so essentially similar DNA fragments will have similar size similar size fragments after being cut by the restriction enzyme. So that's why they can be used to identify each other. Okay, essentially the result that they will both generate will be the same. So we can use that as a comparison to find out against an unknown and the known. So the next one is that DNA fragments have a negative charge. This gel electrophoresis uses that because one side of the gel is negatively charged, the other side is positively charged. And we know that basically different charges will attract each other. So since DNA is negatively charged, it will be naturally drawn to the other side of the gel, which has a positive charge. Remember, negative and positive will be attracted to each other. If it's negative and negative, there will be a repulsion. So that's why the gels are loaded at the negative side and they will travel to the positive side. And this is caused by the phosphate groups in the backbone of the DNA. Remember, DNA, nucleic acids are made up of nucleotides, and nucleotides are made up of 3 parts, right? One is the ribose sugar, second is the phosphate group, third is the nucleotide. I hope you guys still remember what that looks like, right? This is what one nucleotide looks like. These join together to form a nucleic acid. And this phosphate group up here, that is the negatively charged part. So that's why when you put it in a gel, inside is negatively charged, and the other side is positively charged. The DNA will naturally flow towards the positively charged side because there is an attraction of the charges. And just because there's an attraction doesn't mean that will necessarily create the bands. This combined with the next one, which is the fact that these gels actually have pores in them that allow the DNA fragments to travel through them. But there's one condition with these pores, which is that they allow smaller size fragments to get through them faster. So think of it like this. Imagine a very dense crowd and you're trying to get through. If you're a big group, it's always going to be harder for you to get through. But if there's just one person trying to get through that big crowd, it's going to be easier, right? So imagine that's the case with this gel. Since these holes, these pores are so small, they will always allow a smaller fragment. So something like 50 base pairs will always be able to get through much quicker compared to something like 600 base pairs. Because in terms of size, 600 is going to be much bigger compared to 550. So 50 can easily get through all the pores, whereas 600 will take a longer time. The key word here is that it'll take a longer time. Okay, essentially, if you ran this gel long enough, all the DNA fragments will eventually get to the other side because that's one of the factors that influence DNA. Gel electrophoresis is how long you run the gel. So if you don't stop the gel within a given time, it will cause all the fragments to eventually travel because they have all the things that they need. They have a charge which is pulling them. So if they have enough time, there's no doubt that they will get through it. So this factor that the pores are small enough to let shorter fragments through is key for it to separate it based on the size of the fragments. The fact that the heavier fragments, which are the bigger fragments, will be stuck at the top of the gel, whereas the smaller fragments will be able to get all the way to the other side of the gel. Any questions about why the gels can get through faster or not? Everyone clear about that, right? Size of the fragments allows it to travel through faster. Just think of an analogy where you are in a crowded place and there's one of you going through it versus a group of you trying to get through. A group will always take longer and it's always harder compared to one of you trying to get through. So that's the same logic. Since it's a smaller fragment, it will get through faster. The bigger fragments will take much longer to get through. So another thing is that a dye is added to the samples to visualize the bands using UV light, which you guys have already done. And like I said just now, remember that 99% of the genome is very simple and that 1% has high variability. One of the names we have for that is the short tandem repeats. These short tandem repeats are a section of DNA which have a high variability, meaning that in between each person, there's a lot of changes in their short tandem repeats. And we can actually use these sections of the DNA along with gel electrophoresis to differentiate between different people. Remember, short tandem repeats are sections on the DNA. These sections are very different between each other. So they then act as an identification tag for different people. Using gel electrophoresis, we can then see the differences between each sample and how their fragment sizes are, and then compare them to identify people. So I'm going to get you guys to watch this short video on it, because in case my explanation was already confusing.
We'll be talking about gel electrophoresis. What is gel electrophoresis, you might ask? Well, it's a lab technique usually used in the biochemistry lab for separating out DNA or proteins based on their size. And let's talk about how it works. So first you need to have the gel. This can be made out of different kinds of substances such as agarose and polyacrylamide, both of which I'll discuss later. And the electrophoresis part of it means that you need to have an electrical field passing through the gel to get the bands to move. So to create an electrical field, you have to have a cathode and an anode. The cathode is on this side. Remember from general chemistry that at the cathode, reduction takes place, so you'll have a negative charge. But on the other side, where the anode is, you'll have a positive charge because this is where oxidation is taking place. And to power this, you'll need some kind of battery to connect them. And these are both usually also connected to a big box that contains the gel electrophoresis apparatus. In order for charge to flow across, you need something that will conduct electricity. So you have a buffer that's usually composed of different ions. So this is completely covering this entire gel. And remember that it's always there, but I'm going to erase it just because it's going to get a little messy.
All right, in case you guys are already lost in the video, let me catch you up to what's happening. All right, essentially, this is the gel. These are the wells that you guys were loading stuff into. There is a charge that's created where the wells are closer to the negative charge, and the other side of the gel is a positive charge. There is basically a circuit running through it, right? An electrical current. Remember that DNA is negatively charged, so naturally it will want to go towards the positive charge. This is something we want to happen, okay? There's also a buffer solution that's poured across the gel, which you guys also did. This buffer is basically to allow the current to flow, okay? So basically you're combining a bit of physics into biology, but so far that's what it's doing. Let's see what happens next.
So next, you'll load a sample of DNA. In order to load your sample, you'll need to take a pipette and put it into one of these wells. So usually you'll want to make sure that you have a dye mixed in with your sample so that you can see it as it's running. So I'll show that as pink line here. So I've filled that well, followed by yellow here and green here. So that's just the color of the dye. It can really be any color you want. This will just help you track the movement of the bands later on. Once you have these bands, what will happen once you turn on the electrical field? Remember that DNA has a negatively charged backbone because of all the phosphate groups. So what you'll actually observe is that they'll travel towards the positive end. Okay, so maybe after a short period of time, what you'll see is that it'll look something like this. Maybe the pink will have split up into a few bands, indicating that there's a few different size fragments in there. The yellow, you might only see one band. And with the green, you might...
All right, pop quiz. Who have I not called out yet? Yeah, go ahead. Okay, you see these three bands here, right? Which one would be the biggest fragment? Which one would have the biggest fragment in it? The one on the left, center, or right? Left? Why? Can you give me an explanation? You gotta give me an explanation if you have an answer. Okay, think about it. How does the fragments get through the gel? What do they use to get through the gel? There are holes, pores in the gel, right? In between each well, in between each band, there are pores that allow the fragment to travel through. But there's one condition with these pores, which is what? Why is it the heavier fragments will always be at the very start versus the lighter fragments which will go to the other end? There's always a reason for this. Any ideas? Kevin, what do you think? Why? Why is it that the fragments that are 400 or 500 base pairs will appear here, but 50 base pairs will be here at the very end of the gel? It has something to do with the pores that are across the gel that allow the fragments to move across it. Any ideas? Yeah. So essentially, think of the analogy, guys. Think of the analogy, crowded room, you're trying to get across. There's two groups. One is just by yourself. The other is with a group of 5 to 10 people. Getting across will always be harder if you're a bigger group, right? Same thing here. Because we're bigger size, these pores are going to act as a barrier on some level. That's why the bigger fragments will always be at the start. Whereas since the pores allows smaller fragments to get through them very quickly, so the smaller fragments will get through them to be at the very end of the gel. Remember, it is very much a size-based thing. These pores are size-based. They will let the shorter fragments get through. Bigger fragments will take more effort and more time.
Might actually have two bands, but they're so close together still that it's kind of hard to tell. So how can you really see what's going on? The solution is to let this run for a longer period of time. And eventually what you'll see is shown here at the bottom. You'll see that whatever size fragments were in your original samples have effectively split up by their size. And note that in pink, you'll always want to load something known as a DNA ladder. A DNA ladder is like a standard. This is something that you buy from a company and they tell you exactly what sizes their fragments are so that you can match them up to your unknown samples. So this could be 400 base pairs, 200 base pairs, and 100 base pairs. And as you'll note, the smallest fragments travel the furthest. This is because the smallest things are really easy to push with the electrical field. But when you have such a big molecule, or rather a big DNA fragment, it can be hard to move. So you can see that the 400 base pair doesn't move too fast or too far. And what does this tell us about our unknown yellow and green samples? This shows us that the yellow sample has a band that's 200 base pairs long, and the green sample actually was composed of two different fragments, one that was 100 base pairs and another that was 200 base.
So that's how you would use these gel electrophoresis, right? You're basically interpreting the size of the fragments that were generated and then comparing them against the ladder. Okay, the ladder acts as basically a standard of what are the fragment sizes going to look like, because you already know those fragments, right? Yeah, Zoe? Yes. So restriction enzymes cut the sites, and since, think of it like this, you collected a sample. So let's say there's a murder scene. You collected DNA from all the suspects, and then you have evidence which has a DNA sample of who the killer is. Okay, you amplify both samples, and then you use the same restriction enzyme on both samples. And since both samples are essentially the same DNA, the places where they're gonna get cut are the same. So the size of the fragments that are going to be generated are going to be the same. That's why you can actually compare them against with this one, right? Against the ladder, let's say this is your sample, we can tell that whoever this suspect is has a different DNA fragment sizes. So essentially it's not the same person, because if it was the same person, they would have the same size bands. That would be a more conclusive evidence. But since they have different size, even though there is similarity here, but since there isn't any here, that is a sign that it is probably someone else. Okay?
Scarce. So what would we do with this information? If you needed a particular size of DNA, say for the next step of your experiment, if you wanted to insert it into a plasmid or a vector, you could cut this out of the gel and use it for that. Now let's talk about the two kinds of gels that are most
All right. That is unnecessary at this point.
common.
All right. So I know this looks very intimidating, but let's go through it. We've already talked about all the steps. Then after this, it's very easy, right? First thing, we load the gel with the ladder. This is basically a standard that with the fragments that we've already known. They basically tell us these are the sizes of fragments inside this ladder. So all the things that all the bands that it's gonna generate are already known. That's why it's called a standard. Okay. After that, all the other wells are loaded with the samples to the lane 2, lane 3, lane 4, based on the samples we've collected. And these are placed at the negative electrode. Make sure the wells are placed at the negative electrode, because we want DNA to move across the gel, and that can only happen if there is a current going to the positive side. Okay. Next is that the DNA current is run through the gel, where the negatively charged
DNA particles will move across the gel, traveling towards the positive side. And since the pores allow smaller are, I'll say, preferring smaller size fragments over larger size, the smaller size will move across the gel much faster compared to the larger size. And eventually, after a fixed amount of time, let's say 1 hour, the gel is then the current is then switched off. Then the DNA fragments will settle in whichever well they are stuck at. Okay, so if they've already moved across and are at the 300 base pair well, once the current is switched off, all those fragments will just start to settle at that gel, settle at that well, and create a band to be visualized. Remember that by themselves, these bands are not able to be visualized. They need a fluorescent dye, and that's why we use something known as ethidium bromide, which allows us to visualize it under a UV lamp. So you guys did all these steps when you were at when you were at Ecolink, and you then generated a gel with those bands, right? That's how you were reading them at that time. You were basically reading the size of the fragments to then identify it based on the information that you had. So how do we interpret the gel electrophoresis? We've also looked at this. The shorter fragments will appear in the bottom of the band, bottom of the gel, versus the longer fragments which appear closer to the original wells. And these bands are measured either as base pairs or kilo base pairs. So you will see KBP or BP. Remember, those are base pairs or kilo base pairs. And when we're saying base pairs, we're basically talking about when, let's say, A with T, this is one base pair. That's why it's called a base pairing, right? So when it says 4 kilo base pairs, that's basically 4,000 base pairs that are making the fragment. Remember, these are DNA fragments. That's why they will have a base pairing. Okay. Anyone confused about the base pairing thing? No? Okay. Moving on. So you can see here in the picture, the sample starts out with long and small fragments. And once the current is run through, the long fragments will be stuck at the start, where the short fragments will easily get through all the pores, getting to the other side of the gel. Remember, negatively charged, moving towards the positively charged. All right. So let's think about how to interpret this gel. So let's, Ivan, what can you say about sample 1 in terms of the size of the base pairs? Size of the DNA fragments from sample 1. Don't look at the answers, look at the question. Don't look at, but the next slide literally shows the answers. So don't look at that. All right, so what can you say about sample 1? Just look at the bands and the plates and just let me know that. What size are the fragments for the bands? Look at sample 1. Let me zoom in. Maybe that will help. All right. So let me work through sample 1 with you guys, and then maybe you can have a better example of how to interpret this. Okay. So on the first well, we have the ladder, right? These are the ones with fragments that we know the sizes for. That's why we got 500, 400, 300, and 200. We know because the ladder comes with these information, right? And then we put samples into our individual wells. So when we look at sample number 1, what are the sizes of the fragments that it has generated? There's 400 base pair fragments, and there's also something along the lines of 240 to 250 base pairs. I would say more towards 240, but this is a bit of leeway where even if they ask you to identify it, they would give you some leeway, but you have to make an assessment based on where it is in the positioning between 300 and 200. So basically use your math skills to try to guess what this might be. I would say 250 and 400 base pairs, right? So that's essentially how you would answer that, right? You would say the molecular size of the fragments are in 2 bands, specifically 250 base pair bands and 400 base pair bands. And that's it. That's pretty much how you're interpreting it. Once you can do that,
you can move on to the next step of comparing it with other people. But this is basically how you start out interpreting a gel electrophoresis. You identify the bands that it's made, compare it against the ladder to know what are the sizes of the fragments. Okay, next is sample 2. So can I get Emelinda, what can you tell me about the sample 2? What are the fragments? So if you had to say it in the same way as said for sample 1, how would you say it?
There are a lot of size of fragments is 2 bands, yes.
Is it in 2 bands? Look at sample 2. Is it in 2 bands? Okay, so what are the sizes? All right. So yeah, like I said, you have a bit of leeway. Even if you say 150, since there's no clear identification of it, you might have a chance of getting marks. Okay, but try to make an assessment of whether it's at the halfway point between 200 and 100, and then you can kind of guess beyond that. Okay, so last one, can I get? Sorry. All right. What can you say about sample 3?
It's a normal because that's 200.
So the molecular size is 200. Is there anything else that kind of sticks out about it? Yeah. So that's actually another indication, right? The fact that it is a thicker band indicates that there is a greater volume of fragments. So another piece of information that you can derive, for one thing, molecular size, 1 band for 200 base pairs. Okay, that's the important one. The second thing you can make out is only DNA fragments with the size of 200 base pairs were loaded in the well in the first place, because actually no other bands have been made, and the band at 200 is so much thicker compared to any other band that's been formed. So that shows that all the fragments that were put into well 3 were basically 200 base pairs. Okay, these are the kind of information that they are expecting you to be able to derive from a gel electrophoresis, and then you have to use that information to compare against other samples. So, like I said, gel electrophoresis can be ruined by 4 things, right? There are 4 things that can happen that can basically destroy the results of a gel electrophoresis. One is that the voltage. Remember that current is what creates that attractive force for DNA to move across the gel. So if the voltage is somehow not consistent throughout the running, it can ruin the gel altogether. In case if the DNA, if the voltage is suddenly increased too high, then the current is too strong, then all the gel will start running across, all the DNA fragments will start forcing themselves through the gel, so it ruins the data. Next is the composition. This is basically how dense the gel is, which impacts how fast the DNA fragments can get through. Buffer concentration basically controls how much current is then able to pass through the entire solution, which influences the movement of DNA fragments. And then time. Remember, like I said, the gel is not impervious, so eventually the DNA fragments can get through it if you have enough time. So that's why even when we were running even when we were running gel electrophoresis at EcoLink, they were specific about the time, and they were saying that if you had continuously run the gel, all the fragments would actually jump out of the gel itself, falling into the machine. Wish I could have shown you guys a more clearer picture of that. So we do these kind of gel electrophoresis testing. One of the benefits is paternity testing, maternity testing, also for generally DNA, forensics. These are the 3 major uses for gel electrophoresis in this context. So you can see here, you have the mother, which has a specific specific data, a child which has specific bands,
Now you compare the mother child against the possible father. So what you can say here is that the bands that the child has that are similar with the mother can be accounted for by the mother. Basically, it has those bands because of the mother, but the bands that the child has that the mother doesn't have would probably have come from the father. So looking at this picture, which dad do you think is most likely the father? Sorry? The third one. Alright, let's have a look, shall we? So you can see that for this band, the band at the very top, the mother doesn't have it, so the father must be the one to have it. And when we look at that, that 2 and that 3 kind of match. So now let's look at the second one. Second band, the mom is not giving that, so it's probably not the case. Third band, the mother is not giving that, so it could be just another, could be disregarded. This one is accounted for by the mother. This one, these two bands, the child has, but the mother does not. Those will be the key identification tags you compare against these three fathers, right? The one that is the most matching is dad number 3. He has both bands, which are accounted for in the child. So the most likely result is the dad 3. Remember, this is a very small sample size. Imagine your DNA is made up of hundreds of thousands of STRs, the short tandem repeats. This is basically one region of those STR that has been analyzed. So it's generating data to compare, but it's not foolproof because essentially there is some level of uncertainty here. But we can then improve on that uncertainty by using multiple regions of STRs to do a gel electrophoresis comparison. So I know this looks very intimidating, but don't overthink it. These names at the sides are basically the names for STRs, the short tandem repeats. Remember, those are the regions of DNA that are very different between individuals. So by using a gel electrophoresis on those DNA regions, we can see a lot of variability in between each person. So it's much more convenient to identify a victim with a bunch of samples when we use STRs because it's very different between each person. So let's compare here, shall we? So the first STR, CSF1PO. Let's look at the bands that the victim has generated. So if 14 base pairs, these numbers are the number of base pairs, 14, 13, 12, 11, and so on until 6. What are the base pairs that the victim has generated? Okay, can I get the 4 of you to move to the front for a bit? Let's just get this done as quickly as possible. Can you get, yeah, 4 of you move to the front. I'm going to call out a bunch of people to give me the bands as quickly as possible. So you can go through it. Yeah, I should have hidden this as well. That's my fault. Okay, come on. All right. So you can see that the first STR, the victim generates bands at 13 and at 7, right? And then the prime C, you can see bands appearing at 13 and 12. And the 12 is a bigger band and also at 7. So it is a interesting find here. So we can compare the victim slash the crime scene. You can say that there is the victim's blood found at the crime scene as well, because that's why there are similar bands that can be generated. And then the one part of the DNA fragment which is not found with the victim is the one at 12, which is a significant band size. So we compare that against all the samples, and we can see that individual 2 has a matching band size. Okay, so that's for this STR. We then compare against multiple STRs to see whether our conclusion is right. So we see here, victim slash the crime scene. We know that the victim's blood was found at the crime scene. So we are only looking for these 2 bands. And we can see that individual 2 has those 2 bands. Okay, same thing here. Victim versus the crime scene. We know the victim's blood is at the crime scene, so we disregard that band. We are looking for these 2 bands, which we can find in victim 2. So we know for a fact that individual 2's DNA was found at the crime scene, and we're very sure it is
individual two because we have the data from 3 separate STRs to back up that conclusion. And then we can also make other conclusions. For example, if you had actually looked at victim 1 and victim 3, for all the STRs, they are a very high similarity. Only for one of them, there's a little bit of a difference, but otherwise, no, actually for all 3 of them, they're very much similar. So you can say that they're either identical twins or they're the same person. That's one conclusion you can make. Last one, DNA from individual 3 and 4 share at least one SPL could be indication of relatedness. Okay, so there's a lot of information you can generate from this, and that's pretty much the summary. All right, thank you. Have a good day. Go through the questions yourself if you get the chance.